Thank you. Hello students, welcome to Vedantu NEET English. I am Miss Gopika, your biology master teacher. Some of you might be thinking, why has ma'am wore glasses? Is something happened to ma'am's eyes? No students, can you see the similarity? It's the same person, okay? So yes, we are in a new look, we are in a new series launch. So we thought, why not wear glasses? Now I can see everything. Okay, so how are you all doing? Let me know. Yes, hello. You should also put the glass emoji, okay? Send that emoji with the glasses fast, and then we are going to start the class. Okay, some small error was there. Yes, all GMAs are ready. Chalo. Those who did not see my glass look, yeah, it looks nice, huh? See, how is it? Full Talapati vibes, huh? You people should tell me, yeah. Yes, full, yes, new look. I hope the new look is nice. You like the new look? Yes, awesome. So yes, students, we'll start the class. This is all for fun. Okay, so we have launched our RNA series, right? Basically, RNA and DNA, we know that RNA came first, right? RNA came first. So now RNA series is very, what to tell? It's going to be awesome because our YouTube students are very happy in the launch, right? Are you all? Yes. Oh, DNA people also are here. Very nice. Okay. So students, last class, what did we study? We did the introduction. We did the, what is that? We did the 2 kingdom classification, 5 kingdom classification and kingdom Monera, right? This is what we did last class. Today's class, we are going to complete Protista, fungi, kingdom animalia, plantae and viroids, right? Okay. Yes, okay, awesome. All of you are wearing glasses. Now we'll remove the glasses and start the class. I hope all of you smash the like button. If you haven't marked your attendance, it's very bad. Mark your attendance, then only sit in the class, okay? No bunking in the class, like I always say. Now students, the next topic, today's topic is Protista, fungi, plantae, animalia, viroids, virus and lichens. This is our topic. See, this chapter is all about I would say this chapter is all about memorizing. Very less concepts to understand, but a lot to remember, a lot to memorize. So today we are going to do that together. Okay, ready? Yes, I am doing awesome, Angelina. Yes. Okay, let's start, students. So, Protista. What is this Protista? Your Protista is like a person who is not sure if they have to be in DNA or RNA. Because I saw a lot of messages, ma'am, what is the difference between RNA, DNA, what is that, what is this, right? So, a student who doesn't know if they have to be in RNA or DNA is kind of Protista. That means it is a link between your plants, animals and fungi, okay? This acts as a link, okay? A link between your plants, okay, fungi and animals. I'll tell you why did I say this, but you will get to know it soon. Okay. Now, Protista. Students, Whittaker put who and all into Protista. All of you tell me in the chat box. Very good. Already, Vanilla has already answered. Okay. That is all the unicellular eukaryotic organisms. He told, let's do one thing. Let's put them into Protista. Right. He told, let's put them into Protista. So, our main people who are there is all unicellular eukaryotic organisms. Okay, eukaryotic organisms. Now, why did I say Protista is like the students who does not know RNA, DNA, re-NEET, no re-NEET, right? Confusions people. Protista also is like that. A little confused it is because it has some characteristics like plants, some characteristics like fungi, some characteristics like animals. How many of them in the class like that? Please raise your hands. Okay. Oh, hi Rasna. Okay, how many of them in the class like that? Please tell me. Single cell eukaryotes. Okay. So, here what is happening? They are also linking this Monera with the other organisms because Monera is the only one who is unicellular, right? So, this Protista makes sure that it is linking this Monera also with fungi, plantae and animalia. Got it? Yes. Yes. Very easy. Awesome. Awesome. Now, what is the next thing about them? They have nucleus, right? Nucleus is present because obviously eukaryotes means nucleus will be present, and not only that, they will also have membrane bound organelles. Okay, so students, now what is happening? Little bit evolution is happening, right? Membrane bound organelles. Are you all with me on this? Are you all with me on this? Okay. Now what else in Protista? Students, I am going to divide Protista. What am I going to do? I am going to divide Protista. I am going to divide Protista into Cryophytes, Dinoflagellates, Euglenoids, slime mold, Protozoans. Don't take tension. I am going to make sure that you understand every single word in this class. But I want all of you to stay back in the class. Don't leave the class because you have the freedom to live. Stay in the class. The series is only launched for you. The series is only for you. You know, other channels are not taking classes. So this is a good time for you to prepare and be ahead of other people who are sitting and watching news every day. Okay? Yes? Oh, got it? Are you all with me on this? Yes or no? Yes, ma'am. Tell fast, people. Yes, ma'am. How many of you marked the attendance? What is the attendance now? Has it crossed hundreds? Okay. Now, students, the most big thing about this entire your Protista is they have flagella, they have cilia, they have pseudopodia, they have all the kind of locomotory organs. Okay, so in the throughout this Protista, you will see that we talk a lot about flagella, we talk a lot about cilia, we'll talk a lot about pseudopodia. Got it? These are the things that we'll be talking about. Ciliated, hair-like structure, flagella, pseudopodia. These are the things that we'll be talking about. Now, if I have to divide Protista, right? I'll divide Protista. All of you know the division of Protista. Yes or no? Tell me. Ayyo. 1 second, students. Okay, we are going to divide Protista. I'm going to divide Protista into almost 5 of them. What are the 5 of them? Let's see. Protista, I will divide them, okay, into few categories. Right now we'll write the names here, rest we'll write later. Okay, Cryophytes or Chrysophytes. Let's write Chrysophyte. Okay, wait. Phytes, Dinoflagellates. Students, I think I cannot fit it here. 1 second. All of you, whoever learned this chapter should repeat after me, okay? Dinoflagellates, Euglenoids, okay? Slime mold, and the last one is Protozoans. Okay, are you all with me? Very good, awesome, very good. I love the Josh. Okay, very good. Cryophytes, okay, we have Chrysophytes. Okay, so is there Chrysophytes, Dinoflagellates, Euglenoids, slime mold, and Protozoans. So I categorize this entire Protista into some categories. Now, how are these categories taken into consideration, right? How are these categories taken into consideration? See, look here. If you see Cryophytes and Dinoflagellates, they have some characteristic that of plants. Okay, they have some characteristic that of plants, and if you see slime molds, they have some characteristic that of fungi. If you want, you can tell all these 3 has some characteristics of plants. Why am I telling that? Because your euglenoids in the presence of sunlight can do autotrophism. All of you agree on that? Okay, so your 1, okay, 2 and 3 can be very similar to that of plants. That's why I told you they are a link between, right? They are a link between your plants, animals and fungi. So they are very plant-like. And if I see slime molds, they are fungus type, okay? They are fungi type because they, form spores and all that. So they are fungi, okay? They are very fungi-like. And your protozoans, because of their locomotion, they are considered as animal-like. Okay, they are considered animal-like. So you should know this. See, PYQ, many PYQs I have put in this session. So you can see how the questions come every year. Okay, many students actually leave this chapter thinking that I have 4 marks, 8 marks, but now you know, no, every mark is valid. Every mark is a competition by itself, okay? In 1 mark difference, there will be a lot of students, or in say 5 marks, 4 marks difference. One question, let's consider 4 marks difference, lot of variation will be there. So this is how the Protista categories are divided into. Now let's go into each of them, okay? Let's go into each of them. First, you have Cryophytes, students. In Cryophytes, it's actually very simple. It's like a story. Okay, think of it as like a story. Under Cryophytes, we have golden algae and diatoms. Okay, so now the aquatic form, all that I'll tell you later. Okay, 1 second. 1 second, students. Let me just. Did you all mark the attendance? I'm checking your attendance as well as checking if there are anyone talking nonsense in the class. Okay, yes, okay, fine, awesome, good students. Okay, yes, so students, Chrysophytes. Okay, Chrysophytes, we tell faster Cryophytes, okay, but it's Chrysophytes. In Chrysophytes, basically you have to study about 2 people. First is your diatoms. Okay, diatoms, all of you have heard about diatoms, and the next thing that you have to hear is your desmids. Desmids you might have not heard, but you would have definitely heard golden algae. Now, what's so special about them, ma'am? See, the most special thing about this diatoms and your desmids, especially your diatoms, is that the cell wall. So, understand, in your Protista, every category when you're studying, you will study different types of cell wall. Okay, different types of cell wall. Very good. Yes, diatoms and golden algae or your desmids. Okay, very good. Okay, now where are these people seen? Now, if I say normally, they are seen both in fresh water. Okay, both in freshwater and marine. Students, this chapter has lot to memorize. Okay, this chapter is basically memorizing chapter, more than a understanding chapter, a memorizing chapter. So always you should, you know, continue or revise. Okay, always you should keep revising. Okay, got it? Yes, fine. Now look here, diatoms and desmids or golden algae, they found both in freshwater and marine. Now, as I told you, every Protista when you're studying, you'll have to study about the specialized cell wall. Okay, specialized cell wall. Clear? Yes. Okay. Now, what do I mean by cell wall, ma'am? What are you talking about, right? They are, first of all, let me also write a little more characteristics about them. They are microscopic and floats. Okay, they floats like plankton, students, they will float. This is your exact NCERT. I have put NCERT snip as well because in this chapter there are chances that we miss something. Conceptual matter we might talk, but some words we will miss, so that's why. Now the next most important thing, this cell wall, okay, this cell wall is soap box like. Have you all seen a soap box? Okay, the diatom cell wall are like soap box like. This is how we draw and show you, but I'll show you how they generally look like. Students, they look like this. Okay, they look like this. Okay, like this. This is how they will look. That means they are soap box type of cell wall. This cell wall is made up of silica, and this silica will keep settling in the bed of the earth and form the diatomaceous earth. Is this clear to all of you? Is this clear to all of you? Clear? Okay. So this cell wall is a soap box type. Okay, I'm erasing this. Cell wall is soap box. Okay. Basically, they are overlapping, right? They are overlapping with each other and form a soap box like pattern. So cell wall is soap box, but that is not the most important thing. The most important thing is it is made up of silica. Okay, it is made up of silica, and this silica is undestructible. That means silica is one of the strongest materials, so it is not easy to destruct it. Okay, so it is non or indestructible. Okay, destructible, and because of it they will go and keep settling on the bed of the earth. Okay, they will keep settling in the bottom of the earth, of the seabed, I'll write, okay, of the seabed, to form what, students? To form what? All of you tell me in the chats. To form Yes, very good. Diatomaceous earth. Okay, diatomaceous earth. This is one of the things that now also if you just go to Amazon and type diatomaceous earth, you will see a packet. Okay, you will see a packet which actually helps, like they use it, okay, for different things. So this is called as diatomaceous earth. And students, this diatomaceous earth is nowadays used for multiple other things like polishing. Okay, it's used for polishing. Even not only for polishing, they also use it for filtration of the oil. Okay, for filtration as well. Is this clear? Is this clear? Okay, any doubts so far? Yes, I think no doubts. Okay, no doubts. So far good. Okay, diatomaceous earth. So what did we study so far? Under Chrysophytes, we have two categories that you have to study. That is your diatoms and desmids. Desmids are also called as golden algae. Now they are found both on freshwater and marine. They are microscopic and they float. So we tell like planktons. Okay. Now cell wall is soap-like. That means cell wall is basically overlapping. Okay, cell wall is basically overlapping, and because of the overlapping, see, because of the overlapping, they look like a soap box kind of a structure. Okay, like this, a soap box kind of a structure. And this soap box is made up of silica, which does not, destruct itself very fast. So they will go and settle again and again on the seabed to form a diatomaceous earth. Is this clear? Okay, easy peasy lemon squeezy. And I have even put the image, you can see. Okay, I have even put the image. Now, why did I tell cryophytes or chrysophytes are very near to plants? That is because most of them, right, most of them who fall under this category are photosynthetic in nature. Okay, most of them have the ability to do photosynthesis. Okay, photosynthesis. That is the reason why I told they are plant-like. So for everything the question will be asked, why did I tell it is plant-like, right? That question I am answering now. Okay, students, here you can see cryophytes. The image is not clear, but in the PDF the image will be clear. Okay, yes, yes, correct, correct. Avanita, okay, Aviniha, okay, chalo. Next one, Dinoflagellates. From the word itself, something you're feeling, like that. So Dinoflagellates, here mainly you will be talking about flagella, and you will talk about two flagellas. That is the most important thing. Clear? Yes or no? Very good. Many of you are answering, many of you are chumma sitting, okay? Chumma sitting and talking with each other. All of you should answer, then only you will understand. You should keep answering, even if it's wrong or right, it's fine. Okay, we're not here to do that. Now, look here students, this is also mostly marine. Okay, mostly marine and freshwater. Now see, these questions and all won't come, but in case some correct, incorrect questions come, we have to be prepared for it. Now the next thing, again as I told you, the cell wall. Here also the cell wall is like a plate like. All of you tell me, how is the cell wall? Tell me quickly. Okay, they are also plate like. Now that is not the most important thing. They form furrows. Okay, they form furrows, and from the furrows the flagella will be arising. That's what I told you. Dinoflagellae, that means you talk about the flagella the most. Now the next most important thing about these people, they are seen in almost all the colors: yellow, brown, red, blue, green. Okay, they are found in almost all the colors like yellow. Now I cannot go for all the colors, but I'll try 2-3 colors. Okay, green, red. Okay, rest we'll write it with white. Okay, yellow, green, red, blue also we can write, students. We have blue, easier accessibility of blue. Okay, blue, brown and brown. These are the colors that this dinoflagellates come up with. Now you will be thinking, ma'am, wait, ma'am, did I think about red tide? Okay, red tide and all that, right? So again, we'll come slowly into it, but right now understand they are found in almost all the colors like red, brown, green, yellow and students, blue. Okay, red, green, yellow and blue. Is this clear? Is this clear to all of you? Telegram link of Vedantu, NEET English and Biology by Gopika. Now, let's get into the cell wall. The cell wall is very, very easy to remember because students, cell wall is like made of plates. Okay, that means a strong background they are giving, like plates are formed. So, they have proper cellulosic plates. Okay, cell wall are in the form of plates. These plates will keep them very strong. Now, as I told you, these plates are not just covering the entire thing, or it is not like a one structure. They have furrows in between, okay? They have furrows in between, and from this furrows arises your flagella. From the furrows, what arises? Your flagella. Okay, flagella arises. Don't think this is the organism, this is just the furrow. Okay, here you can see some furrows are there. Okay, again, see here you can see one furrow is there, and like this it's going. See, can you see? Furrows are there. So the cell plates does not completely cover. Okay, the cellulosic plates on the outer surface does not completely cover. That means they have small furrows and like ridges. Okay, students, furrows are there from where the flagella will come out. Okay, cellulosic plates. Okay, I don't write cellulosic. Now, flagella, very simple thing that you have to understand about flagella. One will be longitudinal, one will be transverse. One will come out like this, one will come out like this. Okay, so if I draw one, what to tell your Dinoflagellates, let's say I am drawing like this. Students, it comes in different shapes. Okay, now if one is coming like this, one will go like this. Okay, they will be perpendicular to each other like this. Sorry, like this. Okay, one will go longitudinal, one will go transverse. So dinoflagellates, the simple thing is one is longitudinal, one is transverse. Is this clear? Is this clear? Okay, so I'm writing it here. They have two flagellas. Okay, they have two flagellas where one is longitudinal, arises longitudinally. Again, from the furrows, don't forget that you will have questions from it, and one is transverse. Is this clear? Okay, one is transverse. Now, the next thing about it, because they are present in so many colors, okay, because they are present in so many colors, we see that there are red tide formation. That means there is one special dinoflagellate called Euglenoids. Name is very important. That will do rapid multiplication, and it will give us an image of a red tide. That means when the tide is coming, the whole tide will look red in color because of the rapid multiplication of this Euglenoids. Okay, okay, I'm writing it here. So, red tide formation, students, the name is important, okay. Red tide formation is because of your Euglenoids, okay. Euglenoids. How this is formed? Because of rapid multiplication of this, okay. This is formed because of rapid multiplication. Now, the next most important thing about dinoflagellates is that they release toxins like neurotoxins or hemotoxins. What these toxins do is they basically kill all other marine animals. That means the toxins are so poisonous it can even affect humans, but it also kills the marine animals. That is your neurotoxin. You can remember, students, for now. Even if you write toxins, you will get marks because only toxins is mentioned in your NCERT. So, whenever there is any harm for this organism, they will release neurotoxins, which are actually lipid soluble toxins. So, they also harm other organisms as well as for us also. Okay, got it? Very important. Fine. Any doubts? Just look at the board. Okay, just look at the board and just tell me if you have any doubts. Yes, they are also photosynthetic. Okay, they are marine and they are also photosynthetic. That is why, see, students, whichever I told under plants, they are all doing photosynthesis. That is the only reason why I put them under plants. Okay, that is your Cryophytes, Dinoflagellates and your Euglenoids. Somewhere or the other they are doing photosynthesis. That's why I put them under plants. Is this clear? Who asked me? Praveen, are you clear? Yes? Okay, clear. Okay, ma'am, clear. Clear? Any doubts so far? Please ask me. Now, otherwise we will go into Euglenoids. Okay. So, dinoflagellate structure has to be clear. This is very important, transverse and longitudinal. Now, students, next one is Euglenoids. Now, about Euglenoids, if you see, you would have all seen Euglenoids very commonly because from small age you would have studied Euglena as an example. Yes or no? Euglena, you keep telling. Again, same thing, flagella will be there. But the most important characteristic of this Euglenoid is that when in presence of sunlight, okay, in presence of sunlight, they do autotrophic nature. That means they make their own food. Okay, autotrophic in nature. They form their own food. But when sunlight is not there, that means in absence of sunlight, what this fellow will do? this fellow will be like, okay, I'll eat smaller organisms than me. That means it will suddenly become heterotrophic in nature. This is all again like a natural selection, survival of the fittest. It cannot wait for sunlight. So what it will do, whenever sunlight is not there, that means especially during the night times, it will consume organisms which are smaller than them. Okay, it will consume organisms which are smaller than them. This is one of the most important characteristic of it. Now they are found in fresh water, stagnant water. Okay, they are found in fresh water, that is especially in the stagnant water. Now they also have flagella, students. They also have a presence of flagella. One is short and one is long. Can anyone tell me about the cell wall? Hi Chandana, can anyone tell me about the cell wall in your euglenoids? Do we have a cell wall, or something else is replaced, or some protein is there? So here also, students, in this entire Protista, please make a note separately about all the cell walls. Here also cell wall is absent, but they have another, okay, like a replacement. They have a protein, okay, they have a protein layer called as the pellicle, okay, protein layer which is called as the pellicle. So instead of that, cell wall is not there, okay, cell wall is absent, but they have another layer called the pellicle. Very good. Yes, absence of cell wall, but they have a protein rich layer called the pellicle. So here I am writing cell wall. Do you want me to write it down here? Okay, because some students later learn from the notes, they might not understand. So I am writing it here. Cell wall is absent, but they have a protein rich pellicle. Done. Now next one I told was about the flagella. Okay, flagella. Flagella, here also two flagellas are there. One is short, one is long. Easy, no? One is short, one is long. Okay. Now students, here also if you see your euglenoid like this, okay, your euglenoid is like this. One long one will be there, one short one will be there. This is how your euglenoid will look like. Okay. Yeah, mine looks like it's going to target someone, otherwise no. Okay, yes. Rani is talking busily. What happened, Rani? Okay, so now students, this euglenoid, I feel bad for the euglena. Okay, so we'll draw one good one. See, perfect. Okay, this is the long one and this can be the short one, and this is a euglena. Now, the very special characteristics of euglena is that I put them into plants. Ma'am, on what basis you put them into plants? Okay, they do autotrophic nature. But for them to do autotrophic nature, that means they have the same pigment as such of higher plants. Okay, they have the same pigment. Okay, same pigment is present like higher plants. That means your chlorophyll A, like that. Okay, same pigment as higher plants. I'm writing it in green so you understand that's something to do with plants. Got it? They have the same pigment as the higher plants. This is all about euglenoids. Very quick revision. Tell me fast. Okay, tell me fast. That is sunlight. It's in the presence of sunlight, it will do autotrophic nature. In absence of sunlight, it will do heterotrophic nature. That means it will start consuming all the smaller organisms. Then students, it is present in fresh water, stagnant water, and protein layer. That means cell wall is absent, so they have a protein rich layer called the pellicle. Are you all with me so far? Now flagella, two flagellas are there, one short, one long. And why did I put them into plants? Yes, they do autotrophic nature. For them to do that, they have the same pigment as such of your higher plants. Okay, and the example is Euglena. Euglena. Yes. Yes, Praveen. You can say that Euglena is a connecting link between plant and animal, mainly because of this characteristic. Okay, because of this nature, you can tell they are the link between plants and animals. Okay, plants and animals. I have read this in some books, but yeah, since it's not an NCRT, you see, you will understand, no, because autotroph, heterotrophs, both will be there. Chalo, ready? Okay, now let's get, sorry students, next, okay, next, that is slime mold. Now slime mold, I told you, it is like your fungi. That means it does some characteristic like fungi. What could be the characteristic? Can any of you guess and tell me what could be the characteristic that it does like fungi? Maybe it forms spores, or maybe it forms some body, some aggregates. Yes, tell me fastly, what does they do? First of all, simple thing about fungi, they are found very terrestrially, okay. They are seen terrestrially. Okay, they are found terrestrially. And not only that, students, they are heterotrophs. Okay, they are heterotrophic in nature. One of the characteristic of your fungi. They are saprophytic also, parasitic also. Main thing, they fall under that. No, they are heterotrophs. Very good, ma'am. That is spore formation. Okay. Yes, spore formation. Correct. Very correct. Okay. Yes. What are you telling? Correct. Okay. Fine. Now, as I told you, they are also saprophytic in nature. Okay. They are also saprophytic in nature. Now, why did we call them under fungi? Is mainly because they form an aggregate of spores called as plasmodium. What do they form? An aggregate of spores called as plasmodium. See, all of you open your NCERT, you will see a structure like this, and there will be some dots in that. Okay, some dots in that. Those are your spores. Students, please don't get confused with your plasmodium, vivax and all that. This is an aggregate called as plasmodium. Okay, this aggregate is called as plasmodium. That means spores get aggregated, plasmodium. They do this during unfavorable condition and they wait. See, every time conditions will not be favorable. So, when the unfavorable condition comes, they will let this to, mature, form spores, get scattered. They will store them and keep, and the moment the condition gets favorable again, they will scatter this. Okay, they will scatter this. So, this entire your fruiting body, we can tell it as a fruiting body, because spores are there inside this. This will undergo like during the favorable condition. All this is happening during favorable condition. Okay, favorable condition. Otherwise, students, this entire thing will get wasted. No, this entire slime mold, no multiplication will happen, nothing will happen, right? So, during favorable condition, they will form the plasmodium, and when unfavorable condition comes, right? When unfavorable condition comes, they will allow this to form spores and wait for it to get scattered. Got it, all of you? Yes or no? Yes or no? Yes. Okay. So, what will they do? They will wait for it to become fruiting bodies. Okay. Again, all this is just maturing, students. Fruiting bodies, and that's fruiting bodies will start forming spore. Okay. Spore formation will happen, and when the condition get favorable, they will start dispersing. Okay, they will start getting dispersed. That's all. That's all. This is the main reason why I called it under fungi. Okay, why I called it under fungi. Clear? Yes, ma'am. Got it. Okay, clear or not? Yes. Now, one very special thing about this, your entire euglena, is that during the spores, right, when the spore formation, spores are formed, they will have walls. Okay, spores are formed, they will have true walls. They will have true walls which are not present in your adult, that means in your mature ones. Okay, mature ones will not have any true walls, but in the spores, the presence of true wall is seen. This is one extra characteristic of your slime mold. Very important. That means when the spores are formed, there will be a true wall formation. Now, for what is that? For protection, basically, because the spores have to get dispersed, right? If walls are not there, they will get completely damaged. So, during the spore formation, they have walls, but once they mature, right, they will not have walls. Clear? Okay. Yes, they are very, very resistant. For them to become resistant, they are giving this walls. Okay. Like for them to be resistant only, they are giving this walls. Otherwise, it will be difficult. Now, the last one is your protozoans. Students, slime mold understood, euglenoid understood, cryophytes understood, dinoflagellates understood. Please tell me in the chats. Please tell me in the chats if you understood all of it. Yes. The likes are not increasing. Many people are not marking your attendance. I know for sure. Okay. Yes. Now, students, protozoans. Protozoans, we put them into animals. Main reason for them to be put under animals is because of their locomotory structures. Okay, because of their locomotory structures. Who does locomotion? Animals does locomotion. So, their locomotory structure is the main reason to put protozoans, like call them as animals. Now, under protozoans, we have 4 of them. Okay, depending on the locomotion, like what are they using for locomotion? We have 4 of them. Is amoeboid. Okay, first one is amoeboid protozoans. Next one is flagellated. Next one is ciliated. Okay, next one is your sporozoans. Okay, sporozoans. Now, just looking at the names, all of you tell me what is the locomotory organ that they are using. or the structure that they are using? Tell me quickly in the chats. I'm checking your chats. All of you tell me. Yes. Yes. What are the locomotory structures? Yes. All of you faster. Yes. Amoeboid. What will be it? Very simple, ma'am. Amoeboid. Pseudopodia will be there. Don't tell amoeba. I'll hit you people. Okay. Pseudopodia will be there. Some of you will tell amoeba, ma'am. Pseudopodia will be there. Flagellated. Obviously, flagella will be there. Only few of you are answering. Okay. I can see. Ciliated. Cilia will be there. That means hair-like structure. Hair-like structure. Sporozoan. Spore will be there. Spore. Spore, how does it work? Students, spores also scatter and they do the job. Right? They do the job. Very good. Cilia, flagella, pseudopodia. Now, under this also you people know many other examples. Now we will go one each of them, like amoeboid, what is there, flagellated, what is there, ciliated, what is there, and sporozoans, what is there. Main thing for them to be put under animals is simply the locomotion. So the basis of classification, okay, the basis of classification is also based on their locomotory structure. Okay, this is also the basis of classification. Yes. Okay, now we will see NCERT. Okay, we will see NCERT. What is NCERT telling about each of them? Look here, students. NCERT, first we will talk about your amoeboid protozoan. I will teach you, but now let's see this. Flagellated protozoan. These members of this group are neither free living. Okay, this is visible, right? Free living or parasitic. They have flagella. Obviously, you know that. The parasitic form causes diseases such as your sleeping sickness. Okay, sleeping sickness which is caused by your trypanosoma. Now remember this names and keep. I'll ask you questions. Okay, I'll ask you questions. Now, very simple. Only one name you have to study, that is your sleeping sickness is caused by your flagellated protozoan. They have flagella. They can be parasitic or they can be free living. Okay, now ciliated protozoans, they are aquatic, actively moving organisms because of the presence of the hair-like structure called cilia. They have a cavity called the gullet that opens outside the cell. I'll teach you about that. Again, don't take tension. The coordinated movements of rows that causes water laden. Please do understand, all these organisms have this locomotory organs. They are not just for moving. They are also for capturing the food, engulfing the food, engulfing any disease causing anything like that, any other foreign particles, they will engulf them. Okay, they will engulf them. So the coordinated movements of rows of cilia causes the water laden with the food to be steered into the gullet. That means with the movement, it also takes in food. Sporozoans, this include diverse organisms that have an infection spore-like structure. Okay, example is paramecium. I'll show you paramecium and teach you this. Sporozoan, this include diverse organisms that have an infectious spore-like stage. Okay, life cycle. The most one is plasmodium. So don't get confused with this plasmodium and your slime mold plasmodium. Here they are talking about your plasmodium vivax that causes malaria. In malaria, you all know there is a stage or there is a formation of sporozoids. Right, sporozoids, which will be in the salivary gland of the mosquito. It will go and bite the host and then it will get into the blood of the human. All of you remember this? Okay, all of you remember this? Yes or no? Fine. So this sporozoids will go into our human blood and then it will help in the formation of gametes. After that, it will release a hemozoin that will cause us fever. Do not forget, again we have to come to human health. Okay, malarial parasite which causes malaria. Okay, can you all revise faster? Tell me what and all did we study? First we studied about flagellated protozoan. There we spoke about sleeping sickness, which is caused by whom? Which is caused by your trypanosoma. Second thing, what did we study? About your cilia. In cilia, you're going to remember that slipper shaped. Slipper, students, chappal, we say chappal, which we all get hittings from, or your slipper, which is your paramecium. Okay, and nowadays no one gets hittings. Earlier, olden days, our brothers and our parents would have got hittings. Nowadays we don't get. No, sporozoans, again it is because of plasmodium, that is spore formation. Okay, plasmodium, got it? Chalo. Now I'll teach you about amoeboid. Okay, I'll teach you about amoeboid and I'll also teach you about cilia. Cilia, you have to know some diagram. Okay, are you all understanding so far? Slipper like, yes, exactly, slipper like. Okay, Now students, about amoeboid, nothing is there. It's free, fresh water. Okay, they are seen in fresh water and they are free living. Okay, fresh water, they can be free living. The most better thing about them is they have pseudopodia. Not only they engulf, not only they do locomotion, they also engulf food. Okay, they engulf food. And also one important thing about them is the marine form will have a silica covering. Okay, the marine form will have a silica covering. I am writing it here. Marine amoeboid protozoans will have silica covering. Okay, silica covering. So you can imagine how much silica is there in a sea, right? In the sea, in fresh water, so much silica coverings are there, plates are there just to protect them. Okay, this is all. Fresh water, free living, they engulf. Who engulf food? Your pseudopodia or your false feet. They engulf food, they also help in locomotion. Yes? Are we good to go? Yes or no? Fine? Okay, now I'll teach you about your cilia. Okay, cilia, diagram is important. See, I'll teach you about few things in your cilia. Is the diagram clear? Okay. Now students, if you look here, you will see two like a sunflower. Okay, two flowers will be there. One big fellow is there, one small fellow is there. This big fellow is called as macronucleus. Okay, macronucleus. This macronucleus actually does not help in hereditary. Hereditary means basically in your entire reproduction it will not help. Okay, then you will see one more here. Okay, one more here, that is your micronucleus, that means smaller one. Okay, micronucleus. This fellow, even though he is small, he is very active. Okay, very active, very good boy is. He will help in reproduction and also in hereditary. Okay, reproduction, he will help. Because of reproduction, obviously variety will come, right? Hereditary will come. Is this clear? Any doubts so far? Please tell me quickly in the chat box. Okay, any doubts? Fine, no doubts. Okay. Now students, here you see one like a small, like your slipper only it looks, but here you can see one small projection types here. This is your oral cavity. Okay, this is your oral cavity. This portion is your oral cavity. And what did they say? They will bring all the food, and this is your gillet. Okay, through this it will go out. Okay, through this it will go out. So what and all did we study? Macronucleus, micronucleus. This one which you are seeing, this round ones are your contractile vacuole. Again, not very important for you to remember, but if you remember, good. Okay, if you remember, it's very, very good. Contractile vacuole. Now what is happening students? The food will be taken in, through the oral cavity it will come to the gillet, and from the gillet it will be expelled out. Is this clear? Okay. Yes. Yes, macro will help them in metabolic activity or your simple way growth. Micro will help in reproduction and hereditary. This is only for your growth, okay, metabolic activity. And your contractile vacuole is there, gullet is there, oral cavity is there. Now this is the only word according to your NCERT. Other things are just to bring the connection for you. Okay, just to bring the connection for you. Okay, ma'am, clear. Fine. Okay, now notes, everything you will be getting on Vedantu NEET English Telegram as well as on Biology by Gopika. Now, shall we do some PYQs? Shall we do some PYQs? Let's see how many of you understood. Ready? Yes, ready, ma'am. No, ma'am. Yes, ma'am, ready. Okay, chalo. Let's do the PYQ. First question to all of you. I have also asked from Monera. Okay. The primitive prokaryotes responsible for the production of biogas from dung of ruminant animals include. This was asked in your NEET 2016. That time you know, no? Separation was there. So it was asked that time. Tell me the answer. A, B, C, D. Methanogens, eubacteria, halophils, thermoacidophils. The primitive prokaryotes responsible for the production of biogas from the dung of ruminant animals. Yes. You people are singing or crying? What are you telling answer? Some are telling, yeah. How many of you did not mark your attendance? I can understand, okay? Peoples, mark your attendance now, all of you. Those who joined right now, faster. Yes, ma, very good. Methanogens is the answer, because you know methanogens releases methane, and this methanogens are present in ruminant animals like your cattle, cow. Right, from cow you get cow dung that you will use it for biogas. Again, all these topics you will touch a little bit in microbes as well as your human health and diseases. Okay. Chalo, next question. Given figure is of a filamentous blue-green algae nostoc. Identify the parts marked as A and B and select the correct option. Students, this is the A, this is the B. Okay, look at it properly and answer. I hope the diagram is clear. Yes, clear, no? So some questions will be NCERT based, some questions will be PYQ based, some questions will be in-text question. Okay, this is the pattern we are going to follow. So do not forget, you have to answer. See, this is a NCERT based question. That means NCERT diagram, direct question of NCERT. Okay, this is also A. Chumma, don't tell people. It's not A. It's not A. No. Plant kingdom, Basava sir will take. Okay. I'll do animal kingdom. Students, I'm zoology teacher. Why will I take plant kingdom? A for amazing Gopika ma'am. Okay, because you told that, I will agree. Okay. Yes, A is the answer, because this one is your heterocyst. What is the use of heterocyst? All of you tell me in the chats. Heterocyst function. You have to be faster or else I'll only tell the answer. Heterocyst function. Yes, very good. It helps in nitrogen fixation. It helps in nitrogen fixation. They are specialized cells which are present in your nostoc and many other categories of your cyanobacteria, which helps in nitrogen fixation. And mucilaginous sheath covers the entire colony for protection. Okay, mucilaginous sheath or your mucus layer, gelatinous sheath, all these words will be used. Okay, chalo, next question. Dash bacteria oxidize various inorganic substances such as nitrates, nitrates and ammonia and use the released energy for ATP production. They play an important role in recycling of the nutrients. Again, NCERT based question. Every word is NCERT. I just removed one word. Okay, removed one word. Yes, clear? I just removed one word. So by now you should be able to tell. 1 second, students. Just change the AC settings. Yeah. Tell me the answer. B. Okay, many are telling B. Is there any other different answer? 3B. Okay, 3 and okay, B. Okay, fine. That is chemosynthetic autotrophic. Autotrophic, chemosynthetic autotrophs is shown by whom? Shown in Monera. They are the only kingdom that can show chemosynthetic autotrophic nature. Yes? Chalo. Okay, very good. Now let's get back to the class. Again, questions will come after I do fungi. Now students, fungi, we have again some more to understand about fungi. Okay, otherwise it's easy. All of you drink water. Someone tell my name and give, yeah. What you people are like this? No helping nature only. Okay, let's start with fungi. Okay, fungi, what do they do to students? First thing is, as I told you, they are heterotrophic. Okay, heterotrophic. Again, every line of your NCERT will be covered. I have also put NCERT snip. So, we know. Okay, heterotrophic. Under heterotrophic, two types are there. Saprophytic. Okay, saprophytic and parasitic. Okay, saprophytic and parasitic. Now, saprophytic, parasitic. Saprophytic means what? Dead and decayed organism. Parasitic means they live inside. We know that wherever there is some amount of moisture content, that means if the place is a little moist, okay, or damp places, or if the temperature is little warm, okay, temperature is warm, there everywhere fungi will grow as well as bacteria will go. Okay, fungi will grow as well as bacteria will grow. Clear or not? Yes, they are all Gopika ma'am army. I hope now you got the answer. My name is very clear. Okay, they have typed it many times. So students, fungi will be found in all places wherever there is little wetness, dampness will be there. Temperature has to be warm. Okay, that means it has to be a little hot temperature. 30 to 36 degrees is fine. That is the reason why we refrigerate our food. Right? We keep food or anything, we keep it in the freezer, we keep it in the fridge so that no fungi or the bacteria will affect us. Okay, this is basic things about fungi. Is this clear? Now students, I can also say fungi is cosmopolitan in nature. That is because they have a great diversity. Again, you can check NCERT, all these words are from NCERT, okay? Great diversity, and I can also say they are cosmopolitan like your bacteria, okay? Cosmopolitan like your bacteria, because wherever, okay, possible, they'll be there. Wherever possible, they'll be there. They're one problem also, but at the same time, again, many things are being found because of them, right? We are going to study some of the antibiotics and all of that. Yes, are you all with me on this? Yes, awesome, awesome. Now, as I told you, they're not just like, you cannot always say they're the bad people, they're also good people. Why? Because they do symbiotic associations, okay? Symbiotic association with two people, okay? The whole thing is mycorrhiza this is one, okay, mycorrhiza and your lichens. Mycorrhiza is when fungi, okay, comes in contact or stays with your higher plants, in the roots of your higher plants, right? That is where we talk about your fungi. So here it is basically fungi, okay, plus your higher plants, that is in the root nodules of the higher plants. Example, you can think of pinus, okay, example, you can think of pinus. Now lichens, if I talk about what? Lichens is basically algae plus fungi, okay, algae plus fungi. But now what you need to know, students, algal component of your lichen is called as your phycobiont, okay, phycobiont, and the fungal part of your algae is called as your, lichen is called as your mycobiont. Am I right or wrong? All of you tell me in the chats. Am I right or wrong? Yes? Yes, right or wrong? Tell me quickly, fast. Okay, fast, you have to answer. Yes, very good. Okay, so understand that fungi, even though it is heterotrophic, saprophytic, parasitic, they also do symbiotic association. That means they'll help themselves, they'll also help another person. Same way, like both the people are mutually happy. Okay, here also you can see fungi and plants, they're present in the higher plants root nodules. Example is pinus. Lichens also you can see algae is there, fungi is there. Algae will give food to fungi, fungi will do protection to algae. This is the symbiotic, like mutually, okay? Your mom is happy, you're also happy. Your mom is angry, you're also, you're in trouble, okay? So that kind of a symbiotic relationship will happen between these people, okay? Phycobiont is the algal component of the lichen and mycobiont is the fungal component of the lichen. Now what else do we know about fungi? Fungi, what do we know? We know that they're filamentous in structure, okay? They're filamentous in structure, and not only that, we know few more things like, students, I can't move this, no? Okay, we know few more things, that fungi are two types mainly. That is, first one is unicellular and multicellular. Okay, unicellular and multicellular. Unicellular, we have only one hero here, that is your yeast. This fellow was supposed to go to Protista, but then we gave exception and we put them in fungi because we can study them under fungi, okay? Now, multicellular, students, almost everything else comes under multicellular, okay? Now, all the types that you are going to study will fall under multicellular, Phycomycetes, Basidiomycetes, whatever example you are going to study, everything will fall under multicellular because the only unicellular one is yeast. Now, what did I tell you? Fungi is filamentous, okay? Filamentous. Now, you will study more about filamentous. So far, good? So far good. I know the chapter might not be that interesting like a human physiology, but I understand this chapter takes a lot of memory, okay? We forget so many things. It's full of words. It's just words after words. Okay, chalo. Students, when I'm teaching that topic, you people tell. Now only few people tell. Now only they're telling, ma'am, what is that? Your sexual spores and all. Now, let's understand. I told fungi is filamentous. Now, what do I mean by fungi is filamentous? They are formed by a thread-like structure. Okay, a thread-like structure. I will call this thread-like structure as hyphae. What will I call? Thread-like structure as hyphae. Thread-like structure as hyphae. Now, if I form a network, okay, if I form a network of hyphae, I'll call that network of hyphae as mycelium. What will I call it? network of hyphae as mycelium. Network of hyphae is called as mycelium. So we got to know fungi is filamentous, and that filamentous structure is because of the presence of hyphae, and a network of hyphae is called as mycelium. Clear? Okay. Now I can divide hyphae. I can just not be like, okay, one hyphae. No, I can divide hyphae into two types. Okay. Now we'll draw one network. Students, again, network can look different. Okay. Network can look different. It can go from anywhere to anywhere. If you would have seen it, I think you would have done it in your PU. Have you all done it in your PU? You would have done an experiment to see your hyphae. So this will be like a network of hyphae will be there, which forms a mycelium. Now, if I have to divide my mycelium or say hyphae itself, I will divide hyphae into two parts. Okay, two types. Hyphae will be divided into two types depending on septum. Okay, septate and aseptate or cenocytic. Okay, students, these words have to be in your memory. Okay, cenocytic. Now, what do I mean by septate? Very simple. Septum means what? We have studied it in heart. We know that there is a nose. Septum means like a boundary, like a barrier, right? So, if I draw septate, how will it look like? If I draw septate, it will look like this. Okay, it will look like this, right? Septate. That means branches are there or boundaries are there. They are categorized into compartments. But if I draw aseptate, aseptate has no boundaries, nothing is there. Okay, nothing is there. No boundaries, nothing is there. They are just scattered. Okay, they are multinucleated and they are present in the cytoplasm. Okay, they are multinucleated and they are present in the cytoplasm. Whenever they are multinucleated, present in the cytoplasm, and they are like, you cannot properly break them into compartments, you can also say it as cenocytic hyphae. Any doubts? Yes, septate and aseptate, but many places you will see this word. So please understand, they both are similar meaning itself. Multinucleated cytoplasm, we call it cenocytic as well. Clear? Yes, barriers are not present. Very good. Yes. Yes, Savita. Awesome. Awesome. Okay. Chalo. Now we'll try to understand who has what. Okay. Who has what? Now who has what? I have brains. Okay. So now septate and aseptate. Septate is especially seen in higher. Okay. In higher fungi. Now what do I mean by higher fungi? I'll tell you once I finish lower fungi. Okay. Here this is present in lower fungi. Can any of you tell me which is the lower fungi? Like that means primitive. Very primitive kind of fungi. Out of Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes, which is the most primitive one? If you want, I'll give you clue. Okay, if you want, I'll give you clue. Yes, cell wall of fungi is made up of chitin. Okay, chitin and polysaccharide, which is non-cellulosic. Okay, non-cellulosic. Okay, students, look here. The lower fungi is Phycomycetes. Okay, Phycomycetes. Got it? Phycomycetes. And in your higher organism, everything else you put off. Ascomycetes, Basidiomycetes, okay, Deuteromycetes. All these fellows will have septate hyphae. Yes? Yes. Clue I gave now. Full I wrote off. Okay. Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Please understand, when you are writing the characteristics, you have to know who is falling under what. Okay. Yes, their cell wall is made up of chitin. Chitin, they say chitin and polysaccharide, and they put non-cellulosic. Okay, chitin is the main one. Got it? Now this is so far good. Understood? Okay. Now students, under your septate, or sorry, under your aseptate, we can go and divide them further. Okay, we can divide them further. I am doing the division here itself. Now we are going to divide them. One second, students. We can divide this septate into two categories. Okay, depending on how many nucleus is present. Depending on how many nucleus is present. Like monokaryon. Okay, monokaryotic. You can say karyotic or karyon, and dikaryotic. Okay, dikaryotic. Very simple. If I draw a structure, you will understand it in 1 minute. Okay, this is your monokaryon. That means single one. Okay, like this only, single one. If I draw dikaryon, I am drawing the same structure, but I will be drawing, wait, I will draw the, okay, nucleus. You will see that two types are there. Can you see I have drawn it in two different colors, right? Two types are there. Okay, is this clear? Students, you don't go into albedo, that, this and all. I will come into that. Kindly try to understand only this much, okay? That is your hyphae, that is your filamentous structure, thread-like filamentous structure is divided into two types, septate and aseptate. Whenever you hear the word septum, that means a barrier, a boundary, a proper compartmentalization will happen. So septate, you can see proper compartments are formed. This is present in your Ascomycetes, Basidiomycetes and your Deuteromycetes, okay? Now aseptate, it's in lower fungi like Phycomycetes, where entire thing, like in the sense the nucleus is scattered, okay? Nucleus is scattered. Clear or not? Yes? Yes, okay. So that is, they will be scattered everywhere. So this will be simply called as, okay, simply called as multi-nucleated cytoplasm. Okay, multi-nucleated cytoplasm. That means many nucleus are just scattered in the cytoplasm. Now, if I divide aseptate or cenocytic into two types, I'll get monokaryote and dikaryotic. Mono means only one nucleus is there. Dikaryon means two different genetically dissimilar nucleus is there. Okay, genetically dissimilar nucleus is there. Okay, love from me also, from Kerala. Okay. Okay, yes, fine. Here you can see that two different, please do understand the nucleus is that means they are different, genetically they are dissimilar. Genetically they are dissimilar. Now please do understand, from here in your dikaryon stage, students, if there is a region, okay, if there is a state where both the nucleus are not fusing together, okay, both the nucleus are not fusing together, they are called as dikaryon phase. Okay, dikaryon phase, that means both of them will not fuse with each other. That will be called as your dikaryon phase. Again, slowly you will study this. I think DNA students are already doing it now, this chapter. Yes or no? Any DNA students in the class who are doing this chapter? Yes, fast, respond fast. Yeah, you people are very, very slow. Day by day, people are becoming slow and slow. Okay? Yes? Any DNA students in the class who is doing this chapter? Fast, tell me in the chat box. Okay. Now, by the time they answer, we will continue our class. Okay, we will continue our class. Jaldi, smash the like button and mark your attendance. So, is this whole thing clear? Okay, here I will just write about the cell wall, students, because again, when you study, you might miss this out. So, cell wall, okay, is made up of chitin and polysaccharide. Again, they are non-cellulosic, okay, that is the most important thing. Now, let's go into the next part, that is your Phycomycetes. Now, so far good? Did you understand? Now we have to go into each of them, but before I go into each of them, I am going to tell you some more things. Wait, one second. One second. Okay, we will do here reproduction, so the terms are clear for you. Okay, terms are clear for you. Chalo, let's do reproduction. Okay, is this clear? Board is clear? Yes. Okay, chalo, let's do reproduction in fungi. You will understand the different terms that are present. Okay, reproduction. Students, understand reproduction in fungi happens by three methods, that is sexual, vegetative and asexual. Now we'll start with vegetative. Okay, vegetative method, that is generally vegetative reproduction happens because of your binary fission. Okay, binary fission. Others, tell me which are the other ones? Fragmentation. Does budding come into this? Does budding fall into this? All of you, faster. Yes, sexual phase is remaining. Yes, budding also, very good. Budding also fall into this. So this is the vegetative phase. Now the asexual phase, okay, vegetative phase is done. Now your asexual phase includes some of the spores in it. Now why do I say it asexual spores? Is because they are formed mitotically. They are not formed, okay, by meiosis. They are formed mitotically. That is why they are called as asexual spores. Okay, so asexual formation is because of spores like your zoospores, okay, zoospores, sporangiospores, and your conidia, conidiospores. Is this clear? Is this clear? Yes. Facultative is when there is absence of oxygen, they will do they will do carbon dioxide, they will breathe in anaerobically. That means depending on the situation, they will do that kind of adjustment. Okay, chalo, look here. Asexual reproduction is because of spores, that is your zoospores, sporangiospores and conidiospores. Now please do understand, your zoospores, sporangiospores and conidiospores have their own characteristics. So we have to understand their characteristics as well. Okay, like what are the characteristics? Are they motile? Do they have flagella? Are they aplanospores, planospores? So we will make a box out of it. Got it? So chalo, let's get ready with that. But before that, let's also understand the sexual phase. After that, I'll tell you about this spores. Now that you understood vegetative and asexual, let's understand sexual phase. I'll write it here, students. Okay, sexual phase. That means what is happening? What are the spores present? Or is there something else during the sexual phase of this fungi? In the sexual phase also, you will see oospores, okay, or your zygospores. Okay, in other words, you can write zygospores, acidiospores, basidiospores. Students, whom did I miss, students? Oospores and zygospores will go into Phycomycetes. Okay, Phycomycetes. Tell me, whom did I miss? This is under Phycomycetes, this is under acidiospores, this is under basidiospores. Tell me, whom did I miss? Which is the fungi that I missed out of all four of them? Fast, fast, tell fast. Which is the fungi that I missed out of all four of them? Okay, now these are the sexual reproduction. These are the spores that will help in sexual reproduction. Now we also have sexual phase. Okay, that is your plasmogamy, karyogamy and meiosis. Okay, whom did I miss? I think the charts are not moving anywhere here. Okay, tell me. Ascospores have, yes, Ascomycetes have ascospores. Okay, ascospores, basidiospores. Right, who is missing? Yes, who is missing? Fast, fast. Okay, ascospores. Sorry, spelling mistake. Okay, ascospores. Okay, basidiospores. I forgot. Very good, very good. Who is that? I can't pronounce your name, but yeah, very good. Whoever told that, very good. That is, I forgot Deuteromycetes. Why did I forget Deuteromycetes? Because Deuteromycetes sexual phase is unknown. Okay, Deuteromycetes sexual phase is unknown. That is the reason we forgot this fellow. Okay, Deuterofellows sexual phase is unknown. Reproduction, I'll write reproduction, phase will become wrong word. So sexual reproduction is unknown. That means we don't know if there is any spores, any formation, we do not know that yet. Okay, that is why this fellow is called as the imperfect fungi, because its vegetative and asexual phase is known, but their sexual phase is little unknown, or maybe they have put them into some other category. Okay. Chalo, dikaryon phase is when both the nucleus will not fuse with each other and they will stay like that only. Usually they will fuse, but when they don't fuse, they stay like that. It will call as the dikaryon phase. Yes, this is called as imperfect fungi or incomplete fungi. Okay, imperfect or incomplete. Yes, clear? Now we have to learn about the sexual phases. Okay, sexual phases that are present in your entire fungi. Students, now I am introducing you to the words. So when we do Phycomycetes and all, you will understand very fast. Okay, I am introducing to all the words. Got it? So now students, sexual phase. Phase means what? What is happening, right? What fusion is happening? Sexual phase. First sexual phase is plasmogamy. From the term itself, what do you understand? Something related to plasma, right? Plasmogamy. Can you tell me what do you understand from the word plasmogamy? Okay, first is plasmogamy, then comes your karyogamy. Okay, next comes your karyogamy, and next comes your zygote. That is your zygote formation. I'll write zygote formation. Zygote formation. Yes. Plasmogamy, can anyone tell me what do you understand by plasmogamy? Is there any fusion? Is there anything? Yes, there is fusion. Between which plasma? Like plasmogamy, right? Which plasma? Protoplasm, cytoplasm. Okay. Very good. Fusion of protoplasm. Okay. Fusion of protoplasm. Fusion of protoplasm between two motile, okay, two motile or your non-motile gametes. Now please do understand that these gametes are not just present. One cell modifies itself to form a gamete and then they fuse with each other. Okay, now you can see that this is one, okay, this is another cell. Now they do not have gametes. One of the cell, right, one of the cell fuses itself, I mean they modify themselves and do protoplasm, that is plasmogamy, where the protoplasm fuse together between two motile or two non-motile. Okay, two motile or two non-motile. Is this clear? Easy? Yes, right? Easy, right? Now, karyogamy means, students, karyo means what? Obviously, fusion of your nucleus. Yes or no? Yes, please answer. Okay, very good. Yes, very good. Yes, okay. Now, karyogamy means what? Fusion of nucleus, right? Fusion of nucleus. Fusion of nucleus again between two motile or two non-motile gametes. Okay, two motile or two non-motile gametes. Okay, fine. Now, students understand between your plasmogamy and your karyogamy there is an intermediate stage. Okay, there is an intermediate stage. Okay, intermediate stage which is called as your dikaryon stage. Okay, dikaryon stage. This dikaryon stage, as I mentioned again, both the nucleus, that is genetically different nucleus, will not get fused. Okay, two nucleus will be there. Two nucleus will be there like this. See, like this. Let's draw it with two different color. One pink is there, one blue is there, and they will stay like that only. They will not fuse. So that time what will happen? Two nucleus will be present. Okay, two genetically different nucleus will be present. This is called as your dikaryon stage. Okay, dikaryon stage, it's an intermediate stage between your plasmogamy and karyogamy. That means nucleus has to fuse next. So there's one intermediate stage that will stay in some. Okay, and this dikaryon stage is mainly seen in your Ascomycetes and Basidiomycetes. Okay, Ascomycetes and your Basidiomycetes. If dikaryon stage is there, they will form their own fruiting bodies. Okay, Ascomycetes and Basidiomycetes, and if this dikaryon stage is present, they'll end up forming their own fruiting body. Okay, they'll end up forming their own fruiting body. That is the reason why they have ascospores and ascocarp and basidiocarp. Any confusion? Any confusion? A little tricky, but try to understand. Between your plasmogamy and karyogamy, there is an intermediate stage or an intermediate phase where both the nucleus will stay like that instead of fusing. That is called as your dikaryon phase or your dikaryon stage. This is mainly seen in your Ascomycetes and Basidiomycetes. That means when intermediate stage has come. Okay, that will help them to form fruiting bodies. That is the reason why they have ascocarp and basidiocarp. Yes, fine. What is yes, clear, ma'am. All of you, I want everyone to answer, not just one person. Okay, everyone should answer. Fine. If that is clear, zygote formation, that is, students, the last stage is basically your fusion. Okay, fusion of your gametes. Simple, right? Fusion of your gametes to form zygote. Okay, fusion of your gametes to form zygote, and this will happen meiotically. Okay, that is your 2n. Okay, 2 end states. That is, there you studied mitotically. Asexual reproduction spores are mitotic. Here what you are studying meiotically. Okay, N plus N, and this fusion of gametes will form your zygote. Fine. This is your sexual phase. Any doubts here? Please ask me right now. Sexual phase is this. Now we have to study about your asexual spores. Little more detail you have to study, like who is doing what, are they motile, are they non-motile, all this. Okay, very easy, students. Don't take tension. Okay, don't take tension. Once you finish this class, once again if you go read the notes, you will be clear. Now it might be like, oh, I'm not understanding anything, but then you will understand. Okay, slowly you will understand. Again we'll take this chapter, that time you will understand more. Okay, let's divide them. Sorry, 1 second. Yes. Yes. Okay, tell me which are the asexual spores, all of you in the chat. Don't chat with each other. Tell me what are the types of asexual spores I taught you. Yes. Yes, fast. Tell me each of it. First one was zoospores. Second one was, I forgot, so you should tell. Zoospores. Fast, I'm waiting for you. Think and tell me. After zoospores, what did you study? There you studied Basidiospores, Ascospores. After this, what did you study? Yes, Conidio, or before that you have to remember Sporangiospore. Okay, Sporangiospore. And then you studied your Conidiospore. Okay, now let's study if they have flagella, if they don't have flagella, what is the condition. So, zoospores, they have flagella. Okay, flagella is present. Here, no flagella, or flagella is absent. No flagella. Obviously, because this fellow has flagella, I will call it motile, right? Because it can move, I will call it motile. This fellow will become non-motile. This fellow will become non-motile. This fellow also will become non-motile. Now, other than this, there are some more terms. Okay, for non-motile, we also say aplanospores. Okay, aplanospores and planospores. That also we will study. Okay, are you all with me on this? Again, exogenous, endogenous, I'll be telling you. Okay, exogenous, endogenous, I'll be telling you. Before that, try to understand. Since this is motile, we call it planospores. Okay, this will be non-motile, aplanospores. Again, aplanospores. So much to write, no? So much to write, you will get this notes today. Okay, without writing, you will get this notes. Now, understand this fellow will be inside, okay? Endogenous or endospores they are. They will grow endogenously. That means inside the fruiting body they will grow. Okay, this fellow also is endogenously. Conidiospores is the only one that grows exogenously among these three. Okay, exogenously among these three. What do I mean by exogenously? This is your conidia. Okay, this is your conidia. This is your conidia. Okay, conidiospores. Okay, this becomes your conidiospores. Clear? Yes. Planospores. Okay, that also I will tell you. Motile, non-motile, again you will study many places. Plano also can be there, aplano also can be there. That we have to, I'll make you understand. Okay, don't take tension, I'll make you understand. Now, this much clear? This much clear? This is all you have to know. Now, examples again, under sporangiospores, you can study the example of Rhizopus and Mucor. Okay, again you will study it under the classes as well. Okay, Rhizopus and Mucor. Okay, clear? Now, conidiospores is mainly seen in your Ascomycetes and your Deuteromycetes. Okay, Ascomycetes and your Deuteromycetes. Yes, fine. Next one, students. What was the classification based on? Your entire fungi classification, classification of them into Phyco, Basidio, how did this happen? Basis of classification, okay, basis of classification, this question had come last year, okay, 2024. Basis of classification was mainly based on first, what is the first normal thing we classify anything into? Morphology, okay, based on their morphology, okay, based on spore formation, okay, based on spore formation. And the last one, see, first one was morphology, okay, second one was spore formation. Then what will be the third one? Definitely it has to be fruiting bodies, because now you see that you can categorize your Ascomycetes and your Basidiomycetes depending on the presence of fruiting body, that they have ascocarp, basidiocarp, right? So this kind of category was the basis of classification. Now the next word in your textbook, okay, next word in your textbook is oogamous and isogamous. Isogamous, anisogamous and oogamous. What are these terms? Terms are simple, students. I'll draw and show you. Okay, wait, we will move this. Okay, I'm pushing it here. Chalo. Okay, now we are going to learn the types. Okay, that is depending on how the gametes are, you can divide them into 3 types. Okay, gametes, that means gametes, how they look, right? How they look, how are they formed, like that. You divide them into 3. First one is isogamous. Okay, first one is isogamous, next one is anisogamous, anisogamous. Third one is oogamous. Now let's see individually what these people are capable of, or what are these people, right? First one is isogamous. Isogamous. Students, isogamous is very simple. I'll draw a diagram and I'll show you. You will understand next minute only. What does it mean? What does it... Wait, my diagram will make you confused. Wait, I'll draw once again. What does it mean, students? Looking at it, what do you think? Yes, very good. Iso means same. Okay, iso means same. That means both are motile, same size and shape. Okay. Next is anisogamous. Anisogamous. Now I am introducing you to words so that for you the rest of it will be easy. Okay, so please do listen. That is, one fellow will be big, one fellow will be small, but both will be motile. Okay, both will be motile, one fellow will be big, maybe the female is big, male is small. Okay, this is different. Okay, that is different by their size. Okay, different by their size mainly. Now the last one is your oogamous. Okay, oogamous. Now what do you know about oogamous? Oogamous is like in humans, right? One is big, non-motile, one is small, motile. Okay, one is big, non-motile, one is motile. Okay, this is both different in mobility. Okay, different in mobility and size. Okay, and size. Is this clear? Okay, shape also can be different in some. Okay, shape also can be different in some. Is this clear? So these are the 3 types of category depending on the gametes present. Is it isogamous, anisogamous or oogamous? These are the terms that you're going to learn. Okay, these are the terms that you're going to learn. I hope everything is covered. Check your NCERT and tell me if everything is covered. Okay, yes, female is non-motile, male will be motile. Okay, chalo. Now here, students, here one more thing we'll add, that is the most common spore. Okay, this is the most common one. If you see the majority, this is the most common type of asexual spore, that is your conidia. Chalo, now we'll start with Phycomycetes. Okay, Phycomycetes, I have put the snip. So with the snip only we will study the whole thing, because now you know the terms. Now it's only a matter of understanding it. All the terms that will be mentioned here you already know. Okay, chalo, members of Phycomycetes are found in aquatic habitats, okay, and on decaying woods, in the moist and damp places or in as obligate parasites on plants. These are the 3 or 4 places where they are found. Which are the places they are found? We'll write it this side only. They are seen in aquatic habitats. Okay, aquatic habitats. They are seen on decaying woods. Basically, wherever okay, moist condition is there, they are found all those places. Okay, students who are going to stay till the end of the session is going to not even miss one question from this chapter. Okay, decaying woods, that is damp places, and they are also found as obligate parasite. Okay, obligate parasite in plants. Okay, obligate parasite in plants. Okay, now look here. The mycelium is aseptate and cenocytic. This also we studied, right? We studied that Phycomycetes is the only one who has an aseptate. That is your aseptate kind of mycelium. Got it? So mycelium, I am writing it in the side again. Okay, mycelium is aseptate and cenocytic. Okay, cenocytic hyphae is present. Okay, now aseptate and septate can be seen as mycelium as well. Okay, so don't get it confused. Takes place. Now students, look here. Asexual reproduction takes place by zoospores. This also we understood. Now, as I told you, it can be motile or it can be non-motile. Zoospores can see here, it can be motile or they can have aplanospores that are non-motile, because wherever you studied your sexual phase also, you studied both motile, non-motile. Got it? So both will be there. These spores are endogenously produced in the sporangium. Endogenously produced in the sporangium. Understand that sporangiospore also, right? Not only zoospore, your sporangiospore also. Example, what did you study? Rhizopus and mucor. This both fall under your Phycomycetes. Okay, Phycomycetes. Got it? So zoospores, sporangiospores both are present in your Phycomycetes. Now, endogenously produced, a zygospore is formed by the fusion of 2 gametes. These gametes are similar in morphology, isogamous, anisogamous or oogamous in condition. Okay, isogamous, anisogamous and oogamous. Okay, what? Okay, as you say, I will do in my life. Okay, sure. Yes, clear? Got it? Yes, clear? Chalo. Now, students, listen. Some common examples are mucor. See, can you see? That means zoospores and sporangiospores, because here the word came sporangium. Sporangium means what? Inside that who will be there? Sporangiospores. Okay, mucor, rhizopus, and here students, the albugo that you were talking about is a white rust on mustard. Okay, very important. White rust on mustard. Okay, got it? White rust on mustard. Clear? Okay. Now, Phycomycetes, just keep everything in your mind. We will draw a tabular column and understand. But what did we study about Phycomycetes? Their members are present in aquatic habitats, any damp places where it's there, and they are also seen as obligate parasites in plants. Their mycelium is aseptate or cenocytic. Now, what about the asexual spores? Asexual spores are zoospores or sporangiospores that grow inside the sporangium. Now, here if you talk about sexual spores is your zygospore and it is formed by the fusion of gametes. Both gametes can be morphologically similar. That means they can be isogamous, anisogamous or oogamous in nature. Examples are rhizopus, your mucor and your albugo, which is a white rust. Rhizopus is your bread mold and your mucor, you don't have to know anything about it. Are we good? Are we good to go? Are we good to go? Tell me faster. Yes, fine. Okay, fine. Yes. Okay. Now students, Ascomycetes. Okay, Ascomycetes. Now Ascomycetes also I have put the snip. We will learn it from the snip. Look here. Commonly known as sac fungi. Okay, they are known as sac fungi. The Ascomycetes are mostly multicellular. That is penicillium. Okay, or rarely unicellular like yeast. So this is the most important thing is they are called as sac fungi. Okay, they are called as sac fungi. Students, give me 1 minute. 1 minute. 1 minute. 1 minute. Yes, students. Clear? Voice is clear? Okay, chalo. Now, see, the most important thing is who is called as the sac fungi. That is your Ascomycetes. Now, Penicillium multicellular, rarely yeast is unicellular. They are saprophytic, decomposers, parasitic or coprophilous. That means what? They are mainly seen in cow dungs. Okay, they will be seen in cow dung, even they will be seen in your nails and all of it. That is keratinous. Okay, keratinous. Yes, students, with NCERT, what is the use is you will learn NCERT as well. I can write how much ever I want, but you should also know which word of NCERT is important. Now, look here, the mycelium is branched and septate. I'm going to write the important words this side. Mycelium, okay, mycelium is branched and septate. As you know, already know, branched and septate. I hope this is clear. Now, the asexual spores are conidia. Okay, asexual spores are conidia. Spores are conidia. Got it? They are conidia which are produced exogenously on the special mycelium called as conidiospores. This don't take tension, you already know conidia is exogenous in nature. Conidia on the germination produces mycelium. Sexual spores are ascospores. See, this ascospores formation is because of your dikaryon stage, right? Dikaryon stage is there, they can form their own fruiting bodies. Right, they form their own fruiting bodies, and because of the fruiting bodies, this Ascomycetes have their own fruiting body called as ascocarp. Okay, inside the ascocarp, your ascospores will be there, which are produced exogenously in a sac-like asci. Okay, singular. These ascis are arranged in different types of fruiting body called ascocarp. Okay, ascocarp. See, same thing I had written. Any doubts? Any doubts? Tell me fast. Yes, Ram. Yes, yes. Following NCERT is the best thing. Okay, how much ever I write, you should know what is there in your NCERT. Look here, is this clear to all of you? Okay, ascus is there, that means your spores will be arranged and kept inside fruiting body called as ascocarp. Okay, some examples are aspergillus, caliceps and neurosporas. Neurospora is genetically used a lot. Okay, neurospora is genetically used a lot. So what are the examples? Examples, where will we write? We'll write it here on the top. Okay, that is example aspergillus. Students, aspergillus niger, remember aspergillus, calviceps, okay, and your neurospora. Okay, neurospora. Neurospora is genetically used. That means it is used for many genetic works. Okay, many genetic works. Are we good on this? Now, see, look here. Other things also are there, like your murals and truffles. This also is asked almost many years. This question has come about your murals. Okay, murals and truffles. Students, this will be given. If any of you have gone to Korea, you will know when you for ramen, put a very mushroom like black color thing on top of the ramen. Okay, you would have seen it in your K-dramas as well. They put one, they scrape it like this and they add. That is your murals and truffles which are edible. Okay, which are edible. So the question will come, which is edible. Are we good to go? Are we good to go? Yes. Okay. Okay. Done. I think many of them got tired and they ran away. Okay, they ran away. Penicillium also is an example. Yeast also is an example. Write that. Otherwise, go for all of this. Okay, go for all of this. The most important word is sac-like. Okay, most important word is sac-like. Don't forget this word. Got it? Chalo. Next one. Next one. Basidiomycetes. Basidiomycetes, commonly known as, maybe the text is a little small, in PDF it will be visible. Okay, commonly known as, like commonly formed mushrooms, bracket fungi and your puffballs. They grow in soil, on logs and on the tree stumps. Basically terrestrial, very terrestrial they are, and they live on plant bodies as parasites. Example, rust and smuts. Very important, we will be learning that, okay. The mycelium is branched and septate. Students, you already learned all of this, so now it's just basically memorizing, okay, memorizing. That is your mycelium is branched and septate, okay, branched and septate. Now look here, the asexual spores are generally not found, but vegetative reproduction by fragmentation is common. I told you when I added the entire thing in Basidiomycetes. For Basidiomycetes, I did not give any spores. Did you all notice that? Look here, the asexual spores are generally not found. They are not found. That means they do not have zoospores or your conidiospores or your sporangiospores. Is this clear? Any lag is there? Rani is putting lag. Any lag? No lag, right? Okay, students, there's no lag. I can see the live. Okay, that is, Basidiomycetes do not have an asexual phase. Deuteromycetes do not have a sexual phase. Think like this. Okay, they are very sibling like. That fellow does not have a sexual spore. I do not have an asexual spore, like that. Okay, yes. But vegetative reproduction is mainly by fragmentation. The sex organs are absent, but plasmogamy will happen. This is where what I told that time, you should understand that the cells modify itself as gametes and form plasmogamy. That means their protoplasm fuses together because one cell itself modifies. So this is the little interconnection that you do. When you read NCERT by yourself, you will not understand this. That's why you need a teacher's help to understand this. Clear? So look here, plasmogamy is brought about by the fusion of two vegetative or your somatic cell. Is this clear? So what and all did we study here? We studied that asexual spores are absent. Okay, asexual spores are absent. I'm taking this chapter very slow so you understand every single word, okay? Otherwise, I can go faster than this. Now, after that, what did I tell? Plasmogamy happens. Okay, sex organs are not there, but plasmogamy is happening because of because of fusion of vegetative cell. That means cells have modified itself to fuse together. Vegetative or somatic cell. Okay, somatic cell. Is this clear? Okay, very, very important point. These only questions will come. Sex organs are absent. This is what I told you in the starting. Now, look here. Cells of different strains or genotypes. Now, the resultant structure is dikaryotic. This also did I tell you in the class? Okay, this also did I tell you in the class? Yes, okay, that is Basidiomycetes and Ascomycetes have a dikaryotic stage or a dikaryon stage, dikaryotic stage, which ultimately give rise to the basidium, that means your fruiting body. Karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged as fruiting bodies called as basidiocarps. So there we saw asci. Ascis will be arranged in the fruiting bodies to form ascocarp. Here your basidia will be arranged to form your basidiocarp. Okay, I am writing it here. What did they say? After plasmogamy, there will be a dikaryon stage. Okay, there will be a dikaryotic stage. After that, it will be followed by karyogamy. Okay, karyogamy plus meiosis, that is your zygote. Okay, meiosis to form 4. Okay, 4 basidiospores. Okay, 4 basidiospores. That 4 basidiospores are exogenously produced on the basidium. Okay, this will be formed. Okay, exo I am writing here on the basidium. This basidium will be arranged, right? See, look here, the basidia are arranged in fruiting bodies called as basidiocarp. Is this clear? Is this clear? Yes or no? Tell me fast. My oxygen is over. Okay? Basidia, oxygen got over. Now, some common members are agaricus, mushroom, don't forget. This one is very important. Your Eusteligo and your Puccinia. This question will come. Okay, this question will come. The question is, who is your this fellow? Okay, we told in the starting that they can form rust or smuts. See, they will form smut. Students, you can search this in Google. You will see how they are formed on plants. Okay, on wheat plant, on sugarcane plant, they will form red rot, red rust and all that. Okay, and your Puccinia will form your rust. Okay, just search this in Google. I forgot to add the image. You can search this and you will be able to see. Okay, is it clear? You will be able to see. Yes, Puccinia. Okay, yes, fine. Are we good to go? Last one is your Deuteromycetes. Deuteromycetes will be the end of the fungi. Let's drink some water. Yes. Deuteromycetes ready? Chalo, let's do Deuteromycetes. Okay, Deuteromycetes is commonly called as imperfect fungi only because they have their asexual or your vegetative phase, but sexual phase is unknown. Okay, so they have been, first they were moved into different class, then they were not properly fitting, so they were rightly moved back. So like that kind of changes happen. That's why it's called as imperfect fungi. Almost till here, all stories they have written. Okay, now from here, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes. That means Deuteromycetes and Ascomycetes had a very common similarity. So once their sexual phase was known, from Deuteromycetes they moved to the Ascomycetes. Okay. Now the Deuteromycetes reproduce only by sexual spores known as conidia. Okay, conidia. Now, the mycelium is septate and branched. Some members are saprophytic or parasitic, while large number of them are decomposers. Students, this word we are coming across now. Deuteromycetes decomposers. Okay, some of the members are decomposers. They of the litter. That means whatever we whatever waste is there, this they consume the organic matter and help in mineral cycling. Okay, they help in mineral cycling. Some examples are Alternaria. Okay, again Alternaria and Trichoderma you can remember. Okay, Coletrotrichum, it will be difficult to remember. So what are they telling? Sexual phase is unknown. Sexual phase is unknown, or sexual phase is not known. Because of that they were shifted. Okay, unknown. And the next thing that you need to know is they are asexual reproduction, right? Asexual spores are your conidia, which is the most common one. And examples are Alternaria, okay, and your Trichoderma. Again, Trichoderma and all you will be studying in your health, human health and diseases. Is it? Did you, Srinath, searched it? Good boy. Okay, good boy. Is this clear? Students, one more special thing about them. They are decomposers. Some of them are decomposers, right? Some members are, should give respect, okay? Don't tell that fellow, this fellow. Some members you should tell. Some members are decomposers who will do mineral recycling. Okay, mineral cycling. Clear? Okay, done. Deuteromycetes done. Now, your job is to study this properly, okay? Your job is to study this properly. Ready for PYQs? Ready for PYQs? Symbiotic association we did. Mycorrhiza, Pinus and Glomus you can remember for your types, exo and endo. Okay, mycorrhiza, exo and endo, again you will study in some other chapter I believe. I think one of the botany chapters you will study that. Exo and endo mycorrhiza, inside and outside. Okay, so that under endo you can remember glomus and orchid. Fine? Yes. Yes. Yes ma'am, clear. Okay. Ready for PYQ? Ready for PYQ? Okay, chalo, do the first PYQ. It is a PYQ. So, fungi usually stores the reserve food material in the form of, I kind of forgot to tell this, so I put the question here. Now, I'll write it here. Okay, after that we'll come back. Students, fungi So many things about fungi. See, this is why you should put question. They store their food in the form of glycogen. Okay, like animals, they store their food in the form of glycogen. Now the question will be very easy to answer. Yes or no? Right? I told the answer. What is the answer? Yes, glycogen and oil is the answer. Fungi usually stores their reserve food material in the form of glycogen. Okay, next question. Which question? This question. 2024, favorite year of NEET 2024. Right, very good. Many of you answered. Which one of the following is not a criteria for the classification of fungi? If any student make mistakes, beatings, okay, beatings will come. That is, just now I taught you this. I've taught you everything, then only I've put question. Answer now. Answer now. Fast, fast. Those who did not like the session fast. This much you people should do. I'm writing the whole NCERT for you. Yes, what's the answer? 1. 2, anyone is telling? 1. All 1 na? Mode of nutrition, mode of spore formation, fruiting body, morphology of mycelium. Very good. Answer is 1, because this 3 is the main reason for basis of classification we just discussed. Okay, good people. Okay, you got saved by hittings. Now this one also answer. Let me see how many of you remember the examples. If you cannot see, tell me. Rhizopus is there, Ustoligo is there, Puccinia is there, Agaricus is there. Okay, chalo, answer. Answer. If you cannot see, just let me know. I'll read it for you. I finished reading. Rhizopus, Ustoligo, Puccinia, Agaricus. Very easy, no? Come on, fast. Again, NEET 2024 question. Okay, I was solving this during the paper analysis. I think the chats are not loading. Tell it here. 1 is for this, or okay, people are trying to find the answer. Okay, okay. Take your time, take your time. Take your time. I forgot it was a fill in the blanks. Four. Very good. Let's see which are the ones. Rhizopus bread mold, that is A3. Okay, A3, only 2 options are there, 2 and 4. Ustoligo, smut fungus. Okay, Puccinia, rust fungus. Agaricus, mushroom. Okay, so the answer is going to be option number 4. Right, answer is going to be option number 4. Very good. Okay, chalo. Students, now we are going to start with kingdom plantae, kingdom animalia, because they are by itself a chapter. I'm not going to tell anything, but just basic things. Okay, what is plant? Kingdom plantae includes all eukaryotic chlorophyll containing organisms commonly called as plants. That means anything that can do photosynthesis, anything that has chlorophyll as their main pigment will be put under plantae. Okay, a few members are partially heterotrophic, such as insectivorous plants or your parasites. So this is very important. Your pitcher plant, your Venus flytrap, all of this come under insectivorous. That means they take a smaller organism and they consume them. Chalo, okay. Now, bladderworts and Venus flytraps are the examples of insectivorous. Very important. Cuscuta is a parasite. Okay, Cuscuta is a parasite plant. Okay, the plant cells have a eukaryotic structure with a prominent chloroplast and cell wall mainly made up of cellulose. This is the reason cellulose is the most hard to digest, right? That's why animals eat it and we don't eat raw plants. You will study this in the chapter. Now, life cycle of plants has two distinct phases, the diploid sporophytic phase and a haploid gametophytic phase. All of you know, from a sporophyte, a gametophyte will be formed. From a gametophyte, sporophyte will be formed. This cycle will keep continuing, like you saw it in your bryophyte, pteridophytes and all of that, right? Where you have a protonema stage and you have spores forming, then you have your archegonium, antheridium, all of this. Like that, this is what they're talking about, okay? I'm telling about plants. Okay, I hope no mistakes is there, right? Now, look here, the lengths of haploid and diploid phases are whether these phases are free living or dependent on others. Again, alternation of generation, which you study in your angiosperm, right? Your dikaryotic haploid Diploid stage is not there now for you. Diplontic and haplontic stage, that portion is not there for you now. Otherwise, you would have studied alternation of generation. But now you're studying it in bryophyte, pteridophytes also, no? Sporophyte to gametophyte, gametophyte to sporophyte. That's all about your animal, plant. Now your animal, they are completely heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. This is the main characteristic how R.H. Whittaker or all the scientists put the animals and plants. Okay. Chalo, they directly or indirectly depend on plants for food. They digest their food in internal cavity and store food reserves as glycogen or fat. Okay, same as fungi. Now their mode of nutrition is by holozoic. That means we ingest the food, we break it down, then we digest it, right? That breaking down they're telling. And again, we have definite shape, size, higher forms show elaborate sensor and neuromotor mechanism. We are going to start breathing and exchange next class, okay? The sexual reproduction is by copulation of male and female followed by embryological development. Whoever did not, after this session, your homework is to go and like breathing and exchange, or else I won't take class. Okay, breathing and exchange, you have to go like the session and mark your attendance. Okay, why will I do it for free? You people tell me. Okay. Now, students, we are going to talk about virus, viroids and your prion, okay? Prion, however you want to pronounce, you can pronounce. Again, it has many pronunciations. We all know virus is one of the main dangerous reason why we do not get cure fast. Why? Because virus can form its own variants multiple times, okay? Here I have added a paragraph of your new NCERT. Some people might not have this paragraph in your old NCERT, so that's why I've added it this year, so that when I send you the PDF, this is already there. Okay, so look here. Virus means venom or poisonous fluid, and your Ivanovsky in 1897 recognized certain microbes as casual organism of mosaic disease of tobacco. Okay, tobacco mosaic disease. These were found to be smaller than bacteria because they pass through the bacterial proof filters. Then you had in 1898 demonstrated that one more experiment was done depending on your infected plants of tobacco could cause infection in healthy plants, and named the new pathogen as virus and call it as fluid contagium vivium fluidum. That means which can spread, right? And infectious living fluid. Stanley showed the virus could be crystallized or crystal consisting largely of proteins. They are inert outside their specific host cell. Virus are obligate parasite. Like a story, I finished reading it. Now I'll tell you what is the content of this. So students, starting they got to know that something is causing some kind of infection, and when they looked into it, they noticed that they are smaller than bacteria because they noticed that this is passing through the bacterial fillers. Okay, bacterial fillers should stop the bacteria, right? So they were like, okay, something is wrong. Then many other scientists came and they checked that, okay, this is actually causing infection on even, normal plants and everything, and then they came up with the name called as virus. Now virus is also called as your infectious living fluid, okay, contagium vivium fluidum. And now they understood that Stanley was the person who told that maybe outside they have a coating and inside there is something that will get transferred. That is your outside protein coat and inside your genetic material. All of you know your bacteriophage. All of you remember your bacteriophage, right? Bacteriophage is there, like this structure it is, right? This structure. Now in this structure, this is the protein code, and inside one DNA or anything, RNA, DNA, any kind of material is there. This is inactive, right? This is inert. When this will become active? All of you tell me when this will become active. The moment it comes in contact with the host, this will get active and it will release this genetic material into the host. And once it released, the multiplication is so high, rapid multiplication will happen, and because of this, this is very contagious. Okay? Yes? Yes. Got it? Clear or not? So far understood? Okay, this is about virus. Okay, nothing much about virus. You just have to know about their protein code. Now you know that they are made up of capsomeres, right? Small capsomeres. I've added the image. So we'll just write this. Okay, virus could be crystallized. Okay, they have a protein coat. Okay, they have a protein coat. Virus. Okay, I'm talking about virus, not about viroids. They have an outer protein coat, and they have inner genetic material. Okay, inner genetic material. Now this whole thing is inert, right? Their whole thing is inert. They get active, right, only when they come in contact with the host. This is general thing what you know, and viruses are obligate parasites. Okay, they are also obligate parasites. Now students get confused between viroids, virus and prions. So I'll make that classification clear for you. Now look here. In addition to protein, virus also contain genetic material that could be neither RNA or DNA. Again, molecular basis it is. No virus contains both RNA and DNA. A virus is a nucleoprotein and a genetical material is infectious. In general, virus that infects plants have single-stranded RNA, and virus that infects animals have neither single or double-stranded RNA. Is this clear to all of you? What are they trying to say? Very simple. That is virus, okay? Virus that infects plant, okay, that infect plants will have ssRNA, that is single stranded RNA, but if they are affecting animals, okay, if they are affecting animals, they can have double stranded sorry students, or single stranded or double stranded DNA, RNA, or they can have double-stranded DNA. That's all. That's all. Okay. Now, bacterial virus or bacteriophages are usually double-stranded DNA virus. The protein coat called capsid of small subunits called capsomeres. Okay. So they have small capsomeres like this. Okay. Small capsomeres like this. Okay. Forming the capsid. In your textbook it's there, no, green color image. This is your capsomeres, like a peacock I have drawn, but it is not like this beautiful also students, but yeah, okay, these are capsomeres. Together they form the capsid, basically the protein coat. Okay, basically the protein coat. Clear? Yes, capsomeres. So many capsomeres together will form a capsid, that is forms the protein coat. Okay, is this clear? Now, protects the nucleic acid. Why this whole protein is there? Just to make sure that the nucleic acid does not get, you know, lost or anything. Nothing should happen to the genetic material, because that has everything. Okay, it has the ability to manipulate the host nuclei. These capsomeres are arranged in a helical or a polyhedral geometric form. Virus cause diseases like mumps, smallpox, herpes, influenza, AIDS in humans, and it is also caused by virus. We all know many diseases are there, right? Now, your COVID. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing, and vein clearing. So, understand, in plants, to know, okay, if this is, that is, virus is affected or not, it will be leaf rolling, mosaic formation, leaf rolling, curling, yellowing, vein clearing, dwarfing, and stunned growth. Basically, everything, students, everything that can damage the plant will be done. Okay, chalo. Now, viroids. What is viroids? I'll tell you in simple words what's viroids. You don't have to take tension. Okay, viroids are basically protein coat will be absent. In virus, what is there? Protein coat is also there, genetic material also there. In viroids, protein coat will be absent, but the genetic material will be small. Okay, that's all about your viroids. You don't take tension, just know this. Okay, viroids, main thing is protein coat is absent. Okay, coat is absent, but genetic material is present. Okay, but genetic material is present, don't forget that. That means a free genetic material will be present, yes or no? Okay, so there will be a free RNA particle, free RNA particle. Yes, easy peasy, lemon squeezy. Okay, yes, they'll also have low molecular weight. Low molecular weight, they causes a lot of diseases for plants. Okay, now look here, they're smaller than virus, caused potato. Yes, here I'm writing, they cause potato spindle tuber disease. Okay, students, until they cause a disease, we will not know who this fellow is. Okay, so potato spindle tuber disease was caused. It was found to be a free RNA. It lacked a protein coat that is found in virus. So virus, what is there? Protein coat also is there. Genetic material also is there. It can be RNA or DNA. It can be single-stranded RNA, double-stranded RNA, or a double-stranded DNA. Here what is happening? Protein code is absent. Only one free RNA material was there which caused potato spindle tuber disease. Is this clear or not? Yes or no? Okay, PSTD disease was caused, right? And this was found by your T.O. Diner. Okay, T.O. Diner was the person to first ever coin this name viroids or find viroids. Okay, discovery of viroids was by this person T.O. Diner. Now, prion, what is this? Students, the pronunciation can be different. Okay, prions, don't tell crayons, except crayons you tell anything. Okay, because you don't have pronunciation exam, no. So yes, this fellow is totally different. No nucleic acid only. Then you will be like, huh, what are you telling? No nucleic acid means who is going to do the job? Yes, is that your doubt, all of you? Tell me. No nucleic acid. Yes, but how are they causing disease? Doubt is there. Because they have free proteinaceous particle. So, this fellow is KD number, KD of KD, okay. This fellow has no protein, no genetic material, no nucleic acid, right? No nucleic acid, but this fellow has some proteinaceous, free proteinaceous particle. That is more than enough to cause disease. And this fellow mainly affects animals. That person mainly affects plants. Okay, this caused the Cry-Jacob disease. Okay, and also your mad cow. Okay, disease. Yes, mad cow disease and your Jacob disease was caused by this fellow who has a free proteinaceous particle but no nucleic acid. But protein also turned out to be harmful. Yes or no? Okay, protein also turned out to be harmful. This is causing disease only in animals and this one's molecular weight is larger than this one. Okay, molecular weight will be larger. Should I write it with a different color, students? I think this corner page is not seen. Wait, I'll write it again neatly. So much I wrote. So, yes, here I am writing it causes disease only in animals. Okay, and the disease that it caused was mad cow disease. Okay, mad cow disease and your Jacob disease. Again, you will study this in biotechnology. Okay, little more you study this in biotechnology. Is this clear? And their molecular weight is higher than this one. Okay, weight is higher. Clear, students? Yes? Tell me, all of you in the chats. I'm checking your chats. All of you tell me. Yes. Clear? This mainly happens in plants. Okay. This mainly happens in, this causes disease in plants. Yes. Fine? Done? Yes? Everything clear? So much we studied. Everything is clear. Wow, wow. So many NCERT snips have added because you should know that this chapter works only with NCERT. How much ever I write. Okay, some chapters are like this. Now, human physiology, I don't have to put NCERT. Other than that, I can explain. But here, so much is there in NCERT. So, students, this is your bacteriophage. This is your capsomeres together forming your capsid, and this is your RNA material. This whole setup is to protect the genetic material. Okay, are we good to go? Tell me if you have any doubts, and students, go through the notes. If you want to, write something out of it, definitely write out of it and don't make mistakes. Okay, don't make silly mistakes. Clear? Yes, all of you answer. Let me see. I would request all of you, as soon as you finish the class, write a tabular column for Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. When I am doing this chapter again, I will make the box and give you. Okay, but now you have to do it because now that you understood the concept, you have to do that. Okay, again, see, we have written a lot. All these notes you will get it on Vedantu NEET English Telegram. So run and join the Telegram. Okay, see, so much we have written. Got it? Yes. Any doubts? Students, wherever NCERT snip is possible, I've added there so that you understand the snip. And because some of you have the old book, some portions are missing, so I've added that also. And students, if you're starting your preparation right now, the most important thing that you need is a book, right? So I would request all of you to buy Tattva books, because we personally use Tattva books for preparing our notes, and the books are amazing. Every single thing that you need is there in that book. You will not even have to refer NCERT. That much, you know, every word of NCERT is there. Again, additional information is there. So with NCERT, if you see, the entire flow chart will be there. Now the box, as I told you, know, that box also is already there in Tattva. Okay, the link is in the description, so quickly go and buy the book right now. If you have not invested your money into DNA batch, then you should invest your money here, because this is the best option for you to learn. I'll tell you one more use of it. Look here. Now, using the QR code, you can find concept videos of the theory, progress from basic to advanced, and previous year questions. Use the QR code to find detailed solution. That means basically you're getting so many things for this money, right? You have to scan the QR code, you will get a video. After going through the QR code, you will get previous year questions, right? So everything is connected. So make your choice wisely. Okay? Yes. Thank you so much. Do not forget to comment how the class was, and also smash the like button. And if there is any doubts, definitely put it in the comments. And that's it. Thank you so much for joining. See you all. Tata, bye-bye. Don't forget to comment, okay? I miss watching. I'm watching you people. Thank you so much.